Hello and welcome back once again to the Biobasics. This is Dr. Komal Acharya and this time I am going to talk about the cell cycle, that is the series of events through which every single cell undergoes. So let's begin with the cell first. As we know, the cell is the basic functional and structural unit of any living form, including the humans, birds, aquatic creatures, insects, animals, plants, and even bacteria. Every living form is made up of cells except viruses. Structure and arrangement of cells are different in different living forms, including the unicellular prokaryotes, which are made up of the single cell with different shapes and arrangement, while higher organisms, including all the plants, animals, and humans, with complex cellular organization. In this video lecture, I am going to talk about the eukaryotic cell cycle. As far as cell cycle is concerned, the journey of any multicellular organism begins with the single cell that grows, duplicate its genetic material, divide and produce daughter cells. These daughter cells grow further and divide again and so on. Then these cells are arranged according to their role. This is how our body forms and this is called the cell cycle. Scientifically, the cell cycle is an ordered series of events during which the cells grow and duplicate the genetic material and get divided. Basically, the cell cycle of any living form is a combination of four coordinated processes, which includes cell growth, DNA duplication, distribution of the duplicated genetic material to the daughter cells, and the cell division. In case of prokaryotic cell, the cell growth and DNA replication occurs throughout the cell cycle. But in case of eukaryotic cell, this cycle is more complex and divided into two phases. First one is the interphase and the next one is the M phase or mitotic phase. In this video lecture, I am going to talk about only mitotic cell division, but in case of germ cell, the cell undergoes the meiosis cell division also. As far as the interphase is concerned, it is divided in different phases. It's a period during which the cell prepares itself for the cell division by going through these different phases. First one is the G1 phase, which is a period within which the cell grows and the majorly raw materials required for the DNA replication are synthesized. So in this particular phase, cell size increases, cell growth occurs, ATP synthesis occurs, proteins and RNAs are synthesized. In the S phase, which is the phase of new DNA synthesis, the DNA replication occurs, histones are synthesized, and in case of animal cell, the centriole gets divided. Next one is the G2 phase. It's a period between completion of DNA replication and beginning of the mitosis. So all the required raw materials including RNAs, proteins and ATP are synthesized in this particular phase. After completion of this phase, the cell undergoes the mitotic cell division, which begins with the prophase when the cell has highly condensed chromosome. In this stage of the prophase, the nuclear membrane breaks down and the nucleolus disappears and spindle fiber formation begins. In the next stage called metaphase, the microtubules of spindle fiber attach to the kinetochore and the chromosomes shuffle to align on the equatorial plate, which is followed by the separation of sister chromatids during anaphase when the chromatids move to opposite poles. After that, the nuclear membrane reforms and the spindle fiber formation break down. The final phase of the mitosis is cytokinesis, which actually begins during late anaphase and almost completes at the end of telophase, resulting in the formation of two daughter cells. If these daughter cells are to be divided further, they will enter the another round of the interphase. If not, the cell enters into the G0 phase, which is a resting phase or quiescent phase. The cell in this stage remains metabolically active but cannot proliferate further. The cell enters into the G0 stage only when its proliferation is not required right now, or its differentiation is going to occur and it has to play some functional role, or the cell is destined for the programmed cell death. In any such cases, the cell undergoes the G0 phase. The G0 phase is reversible or irreversible phase. The cell in this stage can remain from hours to years until its proliferation is triggered by some stimulus like the growth factor. So these were the different phases of the cell cycle. Now let's talk about the very much important point of this topic, which is how this cell cycle regulates. So for every living form, it's very much essential that each cell progress through this cycle in ordered manner. Right. So the progression through these stages is highly controlled and regulated by a very complicated regulatory system, which involves some security checkpoints that strictly monitor the coordination and ordered manner of each event. Basically, these checkpoints do not allow the cell to enter the next stage until all the events of the previous stage have been completed. For example, the cell cannot enter into the M phase until DNA replication completes, or the cell cannot enter into the S phase of DNA replication until it has grown properly. So let's check these checkpoints of the cell cycle one by one. First one is the G1S checkpoint. It is also known as the start point in case of yeast cell. It controls the progression of cell from G1 to S phase. It checks for the cell growth, availability of growth factor, availability of nutrients, cell size, etc. So if the yeast cell is facing the shortage of nutrients or the cell size is not proper, then the cell cycle will be arrested at this point and the cell will enter into the G0 phase. In case of animal cell, this checkpoint is also known as restriction point. Next one is the G2 checkpoint. Mainly it checks for the proper DNA replication. Apart from that, the production of proteins like tubulin, enzyme production, ATP production are also checked. Next one is the metaphase checkpoint or mitotic checkpoint. It is also known as the spindle assembly checkpoint, as it checks for the attachment of the spindle fiber formation at the kinetochore of the chromosome for the proper alignment of this chromosome at the equatorial plate. Now, apart from ensuring this sequential events of cell cycle, these checkpoints also check for the DNA damage and also function to provide extra time to repair this DNA damage. And for that, there are DNA damage checkpoints which get activated when the DNA is damaged. So there are DNA damage checkpoints in the G1, S, G2 and M phases of the cell cycle, which activated when there is the double-stranded break in the DNA, single-stranded break in the DNA, unreplicated DNA, and even the misalignment of the chromosome. So these were the different checkpoints of the cell cycle. Now the question is that how these events are checked at different checkpoints. So the cell cycle progression and its regulation is managed by conserved protein kinases known as the cyclin dependent kinases, which work together with the cyclin by making a complex known as the MPF, that is maturation promoting factor. More precisely, this MPF is a combination of cyclin dependent kinases, which is protein kinase known as CDK, which performs by the phosphorylation of the target protein, that works in the combination of the cyclin, which is the regulatory protein which is needed for the catalytic activation of the CDK. So this CDK gets activated when combined with the cyclin. Now let's check how this combination works together. So when the CDK gets activated, it phosphorylates the target protein, which is essential for the progression of the cell cycle. Right. So particularly different kinds of CDKs and cyclins are available at different checkpoints of the cell cycle. So basically the appropriate CDK cyclin complex is required to pass through every single checkpoint of the cell cycle. For example, if the cell has to pass through this G2 checkpoint, then it should have CDK1 cyclin A complex. If this complex is not present in the cell, then the cell will be arrested at this G2 checkpoint and it will enter into the G0 phase. Let me give you another example. If the growth factors are available, this particular pathway will be turned on, which is responsible for the synthesis of D-type cyclins. So the cyclin D will be synthesized, it will activate CDK4 and CDK6, and if this complex is available inside the cell, then the cell will pass through this restriction point. Now if the growth factors are not available to the cell, then this particular pathway will be turned off. So the cyclin D will not synthesized, and so the CDKs will not be activated, and thus the complex will be not there. So the cell will be arrested at the restriction point and it will enter into the G0 phase. The cell can re-enter in this G1 phase only when this growth factors will be available. So this was the basic information regarding how the cell cycle works. If you liked my video, please click the like button and share the video. Please subscribe my YouTube video channel. Thank you.